Our next speaker is Steve Munger from the Jackson Laboratory. He's going to be talking about conserved and tissue-specific effects of natural genetic variation on transcript and protein abundance.
Thanks, Fernando. And it's been a long day. I want to thank all of you for staying up to hear my talk. But now just sit back and let me lull you to sleep. This is actually part 3 of what are becoming an interesting group of stories. Part 1 was told earlier today by my postdoc mentor, Gary Churchill. That is a finished product. Part 2 you won't hear about tonight, but it's a really, it's getting more interesting by the second, and that is understanding the aging kidney proteome. And part 3 is really a first draft, and most of these analyses were done in the last week, so it's new to Gary too, so you're going to enjoy this. But it is a very rough draft, but that's what these meetings are for. My slides are available on Speaker Deck. I encourage all of you to post your slides. Okay. You've seen this slide before, but it is my slide. I did make it. And we've, 2 years ago, if you had asked me, told me that proteins were interesting, I would have said no. RNA is the most interesting species in the cell, and I didn't really care about proteins at all, but now I'm starting up a lab and trying to find people that know how to work with proteins, and I'm trying to learn how to do Western blots and IPs, because proteins have become really interesting, and it turns out that we're finding that a lot of the genetic variation that affects transcript levels in the cell do not seem to affect protein levels. And this is the story that Gary talked about earlier. We found that there's a lot of buffering of protein levels. We identified some post-translational mechanisms that we think account for that. And now what I want to understand is how does this genetic variation that affects protein levels, how does it differ between tissues? And so to do that, we're looking at the diversity outbred mice. This is the third time I've seen this slide today. I made this slide, and Dave just told me I made it wrong, which I'm used to hearing from Dave, but thank you. The diversity outbred mice are this incredible reservoir of natural genetic perturbations. And as Gary said earlier, almost all the genes in this population have one or more mutations, polymorphisms, that can affect transcript and protein abundance. They're a very complicated population, and I wouldn't recommend everyone go out and use them. Very challenging to analyze, very challenging to work with sometimes, can be very aggressive, but they're a lot of fun, and they're very powerful. And they have a lot of variation. And so we started with 192 DO mice. We took their livers. We did RNA sequencing. We sent the livers down to our collaborators at Harvard Med, Steve Gigi and Joel Chick, and they did quantitative mass spectrometry. And then we compared the genetic variants that affected transcript abundance with those that affected protein abundance. And we looked in both liver, and then we compared kidney to liver. And Lucy, just so you know, it's Chick Munger et al., not Chick et al. I'm not bitter. But if everyone could just cite it that way, that no. I'm kidding, but really not. Okay, so what we find is that genetic variation affects protein abundance. And as Gary said, we find about 8,000 proteins expressed in the liver, about 3,000 genetic variants that affect protein abundance of 2,500 proteins. You see that a lot of them are cis, so about half of the ones we detect are cis and half of them are distant. When we look in the kidney, and just to remind you, these kidneys are from a whole different cohort of aged mice. So these are not kidneys from the same DO animals that were used in our first study. But what we see is that I didn't copy and paste the same image, but they look a lot alike. We don't see many trans bands. In the kidney, there might be a few small spots on chromosome 9 and chromosome 19 where we might be getting a pQTL hotspot. But again, you see that about half of the genetic variants affecting protein abundance are found close to the protein that's to the gene that's encoding them. So these are local variants. And then another half that lie outside of that diagonal, and these are distant variants. And so we want to compare how are they the same and how are they different. And so we first look at the local pQTL. And if we compare the local pQTL in the liver and the kidney, well, the first thing we can say is that transcriptional mechanisms underlie about 80% to 90% of local pQTL, irregardless of the tissue. So we see this in the liver. Most of them, if you have a local pQTL, you have a local eQTL with the same effect. If you look in the kidney, you see about 80% again. And if you compare the liver and the kidney, what you see is that it's anywhere from 50 to 75% of the local pQTL are also found in the kidney that are found in the liver. And this just depends on how you set your thresholds. But I started looking at these, and then I got confused. And so I want to show you a few examples that have made me question why I do science. Yet I still do it. So DHTKD1, this is a really nice, easy example. It's got this strong local pQTL in the liver at the beginning of chromosome 2. Same in the kidney. We can zoom in on that part of chromosome 2, and what we find is that there are 4 founder strains that share a high-expressing allele, and so that includes WSB129, PWK, and CAST. And if you look in the kidney, you see the exact same pattern, even though we're looking in different sets of animals. And so if you compare these founder strain coefficients in the liver and the kidney, what you see is a really nice linear trend. So this is probably a single variant in a shared promoter or enhancer that's affecting both the liver and the kidney in the same way. That's an easy one. But we also find cases like this. So this is LDHA. It has a cis pQTL on chromosome 7 in the liver, a really nice one in the kidney. When we look in the liver, it's a little noisy, but you can see that the wild-derived strains WSB, PWK, and CAST have a high-expressing allele. We look in the kidney, lo and behold, it's the opposite. There's quite a few of these, actually. And when you look at when you plot the liver versus the kidney founder coefficients at that peak, you see the inverse pattern. And so, okay, that's interesting. Now we get really frustrating, because here's a case where we have a cis pQTL for MESDC2 on chromosome 7. And if you look on chromosome 7 in the kidney, there's nothing there, nothing significant at all. So this is a case where you'd say this is only found in the liver. But when we look at the pattern, what we find is that you have a complex pattern where CAST and PWK are high, NZO and B6 are low. You look in that region here on chromosome 7, and you don't really see anything, right? But lo and behold, if you plot these, what you find is that even though this is not a significant local pQTL in the kidney, for some reason, CAST is high here, PWK, and what you find is that there's really high correlation in the founder strain coefficients, even though that is not a significant pQTL. So this is probably a case where we just don't have numbers. This is a subtle effect, but it's probably real, and if we had higher numbers of animal and a lot more funding from the NIH, we would probably see that this is a significant pQTL. But I'm going to come back to this because I'm finding that a lot of times these subtle pQTLs, I'm becoming convinced that they're real genetic effects. So distant pQTL are abundant, and you can see all the overlap here, except there's almost no overlap. And so again, in the kidney, we find that almost all of these pQTL do not have a corresponding eQTL, so it looks like it's a post-translational mechanism. And if we compare the liver and the kidney distant pQTL, you have about 1,300 to 1,500 distant pQTL in the liver, 1,300 to 1,500 in the kidney, and only about 30 of them overlap. And so these are very different. The distant genetic control of the liver and kidney proteome are very different, very different. And so you saw this earlier with Gary. We used mediation analysis to figure out what are the causal intermediate proteins that are conferring these distant pQTL effects on the target protein. And so what we did is we took that distant pQTL and we conditioned that distant pQTL on the level of protein and the level of transcript for every one of the 8,000 proteins in our data set and every one of the 21,000 transcripts in our data set. And what we found is that oftentimes we could find a transcript or a protein that accounted for that distant pQTL effect on the target protein. And those are causal intermediates. And so here's the case that Gary showed where you have a very strong distant pQTL for TMEM68. The transcript, the local eQTL, does not seem to be affected at the protein level. And when we condition that distant pQTL on these proteins, we find NNT is the only protein where the abundance of that protein seems to account for the distant genetic effect on TMEM68 protein. And you can look and look at the transcript levels, and you find that TMEM68 proteins are more highly correlated with NNT transcript levels than they are with their own transcript levels. And so that's pretty interesting. And so these are cases where we have nice, significant pQTLs, and they were slam dunks, and they got us into nature, and it's been great for me, and it's been a lot of fun. But what's frustrating to me is that we often forget about all these other little signals. And so, we tend to be limited in these data sets. 200 is kind of the max when you're doing proteomics and transcriptomics. And so you'll find these really large local effects, but oftentimes you'll see these, what I call weeds. These are subthreshold peaks that are still, they look like there could be something there. They don't look like they're noise. And so I thought, what if I could use this mediation analysis, and instead of just looking at the significant pQTLs, if I could mediate on all of these weeds? And so I call this pulling the weeds. And so what we do is for each of these subthreshold pQTL peaks, we run mediation analysis, and then, we first condition out that big whopper of a cis effect, and we get that out of our analysis. And then we mediate all the subthreshold peaks. So here I'm focusing on the peaks that are above a LOD score of 5. We do this for all 8,000 proteins. And so you may have 5 or 6 subthreshold peaks for each protein. You times that by 8,000, you're going to get 50,000 or 60,000 of these mediation analyses. And what you can start to do is you can start to sort them based on where those peaks are and what proteins share those peaks. And can you find groups of proteins where you identify the same mediator for all of those proteins? And I'm telling you this because we do. We find this a lot. So here's a case where ProteH2 shares a mediator, DCTN1, with a group of other proteins, including DCTN2, DCTN3, DCTN4. We can take that list of proteins. We can look at the ontology analysis. What we find is you find enriched, this list is enriched for members of the dynein complex, mitochondria, and amine catabolism. That's interesting. You can see there are many proteins that seem to be pretty well correlated with this group of proteins. And you can go further and find evidence in the literature that cytoplasmic dynein regulates the subcellular distribution of mitochondria by controlling the recruitment of the fission factor dynanin-related proteins. So what this is telling us is that even though these are subthreshold pQTLs, we identify a group of functionally related proteins that share the same subthreshold pQTL. For all of those, we identify the same causal intermediate protein that is found in the same functional complex as those. I would argue that even though that's a subthreshold pQTL, it's probably a real genetic effect. And so we can take all of this data, these natural genetic perturbations, and our mediation analysis, and we can build a predictive protein network with that. And in this case, you can actually find co-regulated genes involved in the mitochondrial ribosomal proteins, the regular ribosomal proteins. You can find immunity proteins. I forget what this one is. But what this does is this is basically a hypothesis machine, and we can start to test these hypotheses in the lab. And so that's what I'm doing. And so in conclusion, we find that transcriptional mechanisms are responsible for most local pQTL, but most local variants that affect transcript levels do not affect protein levels. So consider that when you're doing your eQTL study. Half of the eQTL we see do not affect protein levels. Post-transcriptional mechanisms underlie all but a few of distant pQTL, and we find very few distant pQTL shared by liver and kidney. And mediation analysis can identify protein intermediates that confer significant or subtle effects on protein abundance. And I didn't talk about this too much, but it looks like tissue-specific local expression QTL and pQTL are driving most of these tissue-specific distant pQTL. So with that, I just want to acknowledge and thank Gary Churchill for helping me get my own lab. I've worked with a lot of great people. We've had some funding. This is Jackson Lab, a great place to work. It is a great place to work for anyone who isn't going to postdoc with Clement Chow or with, yeah, Joe. Sorry, Joe. It's like, what's the introduction to Joe tonight? Well, we're all mixed up about that. And so please consider coming up to Bar Harbor and doing a postdoc with me. I promise I'll help you get a faculty job. I really will. I will go out of my way. Thank you very much.
Let's thank